Well, hi everybody. We're ready to start chapter 3. So let me begin by sharing my screen and going to the PowerPoint. So what we're looking at here is a composite view of a eukaryotic cell. Now there's no cell that looks exactly like this. Here the artist has sketched in all the possible various organelles that are critical to the life of the cell. You see the nucleus here in blue. Here's the mitochondria that we talked a little bit about recently, powerhouse organelle. Not all cells have these, but some have flagella. That's the plural term. One would be termed flagellum. And the only flagellated human cell is the sperm cell. So I think at some point in your education, you have no doubt heard and learned about these various organelles and sort of the function that they play within a typical eukaryotic cell. So I am not going to spend time now going over the various organelles. I would like you to review the major organelles shown here in table 3.2 of your book. You should be familiar with what the names are, sort of their general shape, what they look like within a cell, and finally, what do they do? What is their function within a cell? So like the organ systems that we didn't spend time lecturing on in class, I'm going to ask you to please go back in chapter 3 and just review these organelles. The chapter does go through each of these and describe them. So what we want to begin with today is this next topic, which talks about the outer surface of a cell. We call it the cell membrane or plasma membrane. And here we see an electron micrograph on the left-hand side of the diagram that has been falsely colored. So to illustrate sort of the relative position of the structure, we see here in yellow the actual cell membrane. So the yellow region, the lighter yellow region to the left of that is the cytoplasm of the cell. That's where the organelles are going to be found. And then to the right of the cell membrane or plasma membrane, shown here in blue, is the outside of the cell. We refer to it as the extracellular side of the membrane. Now, in the early 1970s, scientists began to learn more about what actually the cell membrane was made up of, with the help of electron microscopes and a technique called freeze fracture, where they actually froze the cell membrane and they kind of cracked it in half, and it ended up splitting right down the middle. So what we have here, what we're showing here, are one of the primary constituents of a cell membrane. These are called phospholipids. You might remember reading about them in chapter 2 on the chemistry chapter. They do talk a little bit about that. In fact, in the lower left-hand corner of this slide, which I believe was probably taken from chapter 2, we see here this lipid, a type of fat molecule that has two fatty acid tails. That's shown here at the bottom. This is a phosphate group in light yellow amid another kind of region of the molecule. This is a glycerol molecule, it's called. So these two fatty acids attached to the glycerol, attached to the phosphate group, make up the phospholipid. And we're going to use this particular symbol with a little circle and two tails coming off of it to represent a single phospholipid molecule. And what scientists discovered was that if you look at the quote-unquote head of the molecule, shown here in red, which is this region here, or this region here of the models, it was found to be hydrophilic. Does anybody know what that means? Hydrophilic. Hydro refers to water, and philic means to love. So this is a region of the molecule that likes water. The two tails here are hydrophobic. They fear water. So when you look at their arrangement these phospholipid molecules, if you look at their arrangement in a cell membrane, you'll find that it's in the form of what's called a bilayer, or 2 layers of phospholipids. And that's depicted quite nicely here in the middle diagram, where we have the hydrophilic heads either sticking out of the cell or the hydrophilic heads sticking toward the cytoplasm of the cell, with the hydrophobic tails oriented toward one another. They don't like water, and there's plenty of it outside the cell, and there's plenty of it in the cytoplasm. So the best place to avoid it is to go here in the center. And so those particular phospholipid tails, they orient with one another, kind of sandwiched in the very middle, with the head regions either facing out of the cell, okay, this is the extracellular side, or facing the cytoplasmic side of the membrane. And these, again, are very, very common. These are probably the most common chemical constituent of a cell membrane. And so this diagram on the right-hand side of the figure is, again, an artist's sketch to reveal the phospholipid bilayer, which we can see here. But what we're also introducing now are some other chemicals that are common in cell membranes. And we see these large purple-blue structures. These are proteins. We'll be talking more about those in just a few minutes. Some of these proteins are found on the outer extracellular surface of the membrane, while other proteins, called peripheral, meaning to the outside, some of these peripheral proteins might only be found on the cytoplasmic side of the membrane. And then we have some proteins, like this guy here and this one here, that span the bilayer, don't they? A part of them faces out and a part of them faces in. These are referred to, as the name implies here, transmembrane. They span the bilayer. Okay, again, different from those peripheral proteins that are found either facing the outside or facing the inside of the cell membrane. In addition to those peripheral and transmembrane proteins, we also see here in green another chemical. These actually are carbohydrates. They're sugars. And some of these sugars, the prefix glyco refers to sugar, some of these sugars actually attach to tails of phospholipids. So instead of having a head, this particular molecule would have a glycolipid region and then 2 tails, which you can't see here, sticking down inside. So that's one place you find these carbohydrates kind of sticking out all by themselves. So here's one here as well. But you also tend to find these carbohydrates often attached to proteins, like we see here and here and here and here. We would refer to this particular type of protein as a glycoprotein, meaning it has a carbohydrate component to it. And a lot of times, these glycoproteins are transmembrane proteins. They span, again, the entire bilayer. And I guess I should also mention another constituent of cell membranes besides the phospholipids and the proteins and the carbohydrates, and those are these little bitty yellow guys, which you might almost miss if you hadn't seen a little line pointing to them. These are cholesterol molecules, which is a type of lipid, and it's been found that these cholesterol molecules help in the maintenance of fluidity within the membrane, meaning they assist in keeping this membrane fluid and dynamic. In fact, this is sometimes referred to as the fluid mosaic model to explain the structure of the cell membrane. The membrane is not static or rigid. It is a fluid structure. These phospholipids are moving around here in the bilayer, and these proteins can be thought of sort of as icebergs floating in this ocean of phospholipid. So use that analogy, if you will. Iceberg proteins. Again, some found across the membrane and some found just on the outer surface or on the inner surface of the bilayer. We often talk about the cell membrane having the primary function of being selectively permeable. What this gets at is that it regulates what can enter and leave the cell. That's what selectively permeable means. Certain things are allowed through, in and out, while other things might be restricted. And this often boils down to the chemical properties of the substance that's trying to move across the membrane. And what we've discovered is that some compounds or molecules, like oxygen gas or CO2 gas, and even some steroid hormones, are able to easily pass through the phospholipid bilayer. And that's because they are soluble in lipid. They're dissolvable in lipid. Remember, these phospholipids are indeed fats. They're lipids. So if you are lipid soluble or fat soluble, you can move across the bilayer very, very easily. However, if you're lipid insoluble, if you can't dissolve in lipid, then the only way you can get across the membrane is by making use of these integral transmembrane proteins, those that span the entire bilayer. And I give you some examples here of some lipid insoluble compounds. Amino acids, you might remember from chapter 2, are the building blocks of proteins. Nucleic acids would be things like DNA and RNA. And as we'll talk about later, certain charged ions like sodium ions and potassium ions, chloride ions, any compounds that have a charge, a minus or a plus charge, typically are lipid insoluble and can only get across the membrane with the help of these bridge proteins here, these transmembrane proteins. This next slide goes into a little more detail about the various types of peripheral proteins. Remember, these are facing the outside or the inside, as opposed to the integral proteins that are spanning the bilayer. So again, we've talked a little bit about these guys acting as bridges, right, for the lipid insoluble compounds to get through. What do the peripheral proteins do? Well, here's an example of 4 different proteins on the surface of the cell membrane, or on the outer or inner, I should say, surface. Typically, I think of a lot of these as outer surface peripheral proteins. The fact that sometimes these can help reactions proceed more easily and quickly. We'll talk more about enzymes coming up, I think, in chapter 4. They help reactions go faster, more efficiently. Some external peripheral proteins will help identify that particular cell as part of the organism. And this really gets at the immune system, the fact that, for example, our white blood cells that are present to help recognize foreign objects, foreign things like bacteria and virally infected cells, that they're going to identify those as non-self and gobble them up and phagocytize them and destroy them. And one way that they do that is by looking for, detecting really, these surface proteins that some cells like cancer cells would have, or bacterial cells would have. All cells have surface proteins, but the white cells are able to distinguish between self cells, in other words, other cells of the body, from what we call non-self cells, things that we need to remove: cancer, bacteria, virally infected cells, those sorts of things. So these cell surface proteins play a super important role in helping to label that cell as being part of the organism, part of you. We would call it, again, a self cell, as opposed to a non-self cell, something that could cause harm, could cause disease, things like cancer cells, bacteria that can cause infections, and cells that have been infected by viruses. Those would be non-self entities. And then this last category, cellular adhesion molecules, as it mentions here, will help cells stick to one another. This is especially important in certain types of tissues to help cells kind of maintain some structural framework. They're like the glue that helps hold cells together and actually helps to layer them. We talked a little bit about the fact that cell membranes are selectively permeable, that they regulate what goes into and out of cells, right? And that in turn is based primarily upon the chemistry of the substance that is trying to be moved into or out of the cell. So we're going to be talking next about these 2 broad mechanisms that sort of describe how certain compounds move across the membrane. One's referred to as passive. These four first terms are passive mechanisms, passive in the sense that it doesn't cost the cell any energy for these processes to take place. Remember, we mentioned the other day that the mitochondrion is the powerhouse organelle of the cell. You probably learned that back in high school. And the primary energy currency that cells utilize is called ATP, which stands for adenosine triphosphate. We'll talk more about that in chapter 4. But it doesn't cost the cell any ATP or any energy if we talk about these processes, which we will in some detail in just a moment. Compare these four to the next four, which we say are active mechanisms, and as the name sort of implies, passive, no energy needed. Active, yes, it does require energy. The cell needs to expend its ATP in order for these processes to occur. So if you get confused as we go through these mechanisms, you can always come back to this slide and get the broad overview again, that half of them will be sort of free to the cell, doesn't cost the cell any energy, while the others do require some ATP. So let's talk about the first passive process. You've all heard of it. It's referred to as diffusion. So here we're talking about atoms and molecules and sometimes ions moving from a region of higher concentration to a region of lower concentration until an equilibrium is reached. And the example that I'm showing you here, of course, is what happens if you take a sugar cube and you place it into a beaker of water. Okay, what happens to the sugar as it dissolves? And that's what's going on, of course, as we go from 1, 2, 3 to 4. As time goes on, that sugar cube dissolves as a result of diffusion. The sugar molecules begin to move from a region of high concentration into a region of lower concentration, which would be, of course, the water surrounding that initial cube that you dropped into the beaker. As time goes on, if you could somehow color those little sugar molecules, those glucose molecules, after an hour or two, theoretically, they would be uniformly distributed within that beaker of water. In other words, we would have reached what's referred to as chemical equilibrium, where all of those sugar molecules are evenly spaced out. They've completely diffused throughout the column of water. Let's watch a video on this.
Molecules dissolved in a solution are in constant random motion due to their kinetic energy. One result of this motion is that dissolved molecules become evenly distributed throughout the solution. This tendency of molecules to spread out is an example of diffusion. But how do these molecules come to be evenly distributed? Let's start with a beaker of plain water. What will happen if we now add a lump of sugar to the water? A lump of sugar is composed of many individual sugar molecules, and even as a solid lump, the individual sugar molecules are in motion. When the lump is dropped into the water, it begins to dissolve. Individual sugar molecules move randomly and constantly from the area where they are common to the area where they are scarce. This type of motion, when molecules move from areas of their higher concentration to areas of their lower concentration, is called diffusion. Diffusion continues until all the sugar molecules become evenly dispersed throughout the beaker. The rate of diffusion is affected by temperature, size of molecules, and the steepness of the concentration gradient. Although not specifically shown in this animation, this is one of the processes whereby materials are exchanged between a cell and its environment.
Okay.
Molecules dissolved in...
Now, that example that we just talked about was what occurred when you just put a lump of sugar into a beaker of water, right? Let's now, as it mentions here, introduce the concept of a selectively permeable membrane down the center of that beaker. And so that's what we've done pretty much here, right? We've got this membrane that is permeable to both water molecules and solute. Now, the solute is the substance that's being dissolved in the solvent. Typically, when we talk about aqueous solutions in our bodies, the primary solvent in our body is water. So just think of water as the solvent. Solutes, again, are dissolved substances. And for our argument, or our example, I should say, for argument's sake, let us pretend that these red solute molecules are sugar molecules, okay, that we stole from the sugar cube after it was dissolved. So what we've got in the left half of this beaker, in half A, if you will, is we have some water molecules shown here in blue, and we have a lot of solute molecules, sugar molecules, shown in red. On the other side of the permeable membrane, we have strictly water molecules, don't we? Now keep in mind, this particular membrane is permeable to both water and solute. So the question is, where will the solute molecules move and why? Well, based upon our definition of diffusion, diffusion says that the solute molecules should move from an area of higher concentration to an area of lower concentration until an equilibrium is reached, right? And if you look at what happens as we go with number 1 and then proceed to 2 and proceed then to 3 as time goes on, look what happens to the solute, the sugar molecules. They pass through this permeable membrane, moving to a region where they are less concentrated. In fact, there's none here initially, but as they move across, the red little balls, the sugars, they'll continue to move across into half B until what happens? Until we have an equal concentration of sugar on the right side of the molecule as we do on the left. Now, at the same time that the sugar was diffusing from A to B, where was water diffusing? Well, again, applying our definition, water should diffuse from higher water concentration to lower water concentration. So there's more water molecules concentrated here in the right half, B, than there is in A. So this is depicting the movement of the water molecules across that permeable membrane into the left half of the beaker until we have an equal distribution of the water molecules, shown here in blue, in A and B. So you can have diffusion taking place simultaneously to different substances, assuming, again, you have a permeable membrane that permits those substances to move across. Those processes, those diffusion processes, will occur until we reach, again, an equilibrium of the substances. This same process, diffusion, is what occurs in our bodies as we speak. Right now, it's taking place in you and me. And this gets at how does oxygen and carbon dioxide move from the blood into our cells or from our cells into the blood. You know that the blood is what transports oxygen. We'll be talking more about hemoglobin, that particular pigment found in red blood cells when we get to the blood chapter, but this is an important pigment that oxygen binds to. So in your bloodstream, you have lots and lots of red blood cells. And when that blood is coming from the lungs, it's rich in oxygen. It's very bright red. And when it gets to the heart pumps it to the rest of the body. And when that blood gets to your tissues, it gives up a lot of that oxygen. It does so because of the fact that we have a high oxygen concentration in the blood compared to the cells, and we're using here an example of a skin cell, where there's a lower oxygen concentration because the cell has been utilizing oxygen, which in a process called cellular respiration that we're going to get into in chapter 4. So there is a lower O2 concentration in the cell as compared to the blood. Therefore, where will oxygen move? Well, it will diffuse across this blood capillary into the cell until some sort of equilibrium takes place. Then the oxygen transport will stop. But if you look in that cell, not only does it have a lower oxygen concentration than in the blood, it has a higher carbon dioxide concentration than in the blood, again, due to what's called cellular respiration. This is a substance that builds up as a waste product, a chemical waste product. So we had better be able to get rid of that carbon dioxide, and we can thanks to diffusion, because this gas will move from a higher concentration to a lower concentration, again, until an equilibrium is reached. But that's enough to get rid of a lot of that CO2 from the cell, bring it on board the blood, transport it back to the lungs, and then of course you exhale that, don't you? A lot of the CO2 that you're building up in your tissues, you get rid of that via expiration, in the same way that you gain that oxygen as a result of inspiration. So movement of gases, again, critically important in the maintenance of homeostasis in you occurs thanks to diffusion. This passive process doesn't cost the cell anything. Here's another type of diffusion, which again is passive, called facilitated diffusion. What makes this different is the fact that we are employing now one of those integral transmembrane proteins to help shuttle that substance from a higher concentration into a lower concentration. So to set the stage, here's our phospholipid bilayer, here's the inside of the cell, here's the outside of the cell. Here we have a transmembrane protein, we have a couple of them here, and we're talking here about a substance, and we'll just pretend we're gonna reference the gray spheres here. We'll pretend that those are some sort of an ion, perhaps, or it could be an amino acid. Remember, these are lipid insoluble, so they can only get across the membrane by being assisted with the help of the transmembrane protein. They cannot move through the bilayer like oxygen and CO2 can. So if we have a higher concentration of a particular substance out here, again, let's assume it's an amino acid of some sort, it's going to try to diffuse into the cell where it's less highly concentrated, lower concentration of that substance. And so what happens is it actually binds to a particular protein, as we see has occurred here. That protein shuttles that substance from the outside into the cell. So this is a protein carrier molecule that shuttles just this substance. And that shuttling, if you will, of the substance continues until the concentration is equal across both sides of the membrane. Okay. So what's really fascinating is the fact that many of these substances out here have specific transport proteins that they make use of to get into the cell. Or it could even be moving out of the cell too, depending upon what the substance is. But the key takeaway is this is movement of materials from high to low concentration, but with the help of a protein. Let's watch this video.
In the process known as facilitated diffusion, a special carrier protein with a central channel acts as a selective corridor, which helps molecules move across the membrane. These special carrier molecules that form the protein channel bind only to a specific molecule, such as a particular sugar or amino acid. Once the molecule binds to the carrier protein, this protein helps or facilitates the diffusion process by changing shape and moving the molecule down its concentration gradient through the membrane into the cell where it is released. Facilitated diffusion is similar to simple diffusion in that both involve movement of molecules down their concentration gradient, and this movement is carried out without any input of energy. However, in facilitated diffusion, the movement of molecules will only take place if it is facilitated or helped by a special protein carrier in the membrane. Facilitated diffusion can occur in either direction depending on the concentration gradient. If there is a higher concentration of the particular molecule inside the cell, the same carrier protein would then transport the molecules out of the cell.
Okay. Osmosis, another passive process. Here, we're specifically talking about the movement of water across a selectively permeable membrane. This movement of the water across this membrane that is going to restrict the movement of some things but allow the movement of other things occurs when water moves from higher water concentration to lower water concentration. Now you'll note that I placed in brackets here the term solute. So I'm going to redefine osmosis as the movement of water across this selectively permeable membrane from a region of high water concentration or lower solute to a region of lower water concentration or higher solute. Now, again, a solute is a substance that's being dissolved. So water is always moving toward a higher solute, toward a lower water concentration. Again, this is passive. This does not cost the cell any energy. Here's how we can demonstrate this process. Here's another beaker. Note, however, that this membrane that we have put in the center of the beaker is not just permeable, but selectively permeable. This means that certain substances like water are going to be able to move across, but bigger substances like protein will be restricted or prevented from crossing the membrane. This all has to do with how big the pores are here and the chemical size of these different substances. Water is a very, very small little molecule, it's H2O, right? Proteins are made up of hundreds of amino acids. So this is a very, 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 very large molecule compared to a little water. So in this initial scenario, in part A of the beaker, we have both water and protein, right? Much more protein in A than we do in B. But B has some protein molecules, shown here in purple, and a lot of water. If we let osmosis occur, water is going to move from a region of higher water concentration, lower solute, and indeed that is the case. Look at B, there's a lot of blue spheres here and just a few protein molecules. The protein would be the solute. And that water is going to move toward a region of lower water concentration or higher solute. There are many more protein molecules here in A and many less water molecules as compared to the right-hand side, B. And so protein is not going to be allowed to move. We're restricting it because it's such a big molecule. But the water molecules, they can move across with no problem because they're small. So where will water move? Well, you can see that water is going to move from B to A. It moves from a higher water, lower solute, to a lower water, higher solute. The effect would be literally to cause the level in A to rise due to the influx of water, and the level in B to fall due to the loss or efflux of water from B to A. Again, this is passive, does not require any energy, but we're talking specifically about the movement of water across a selectively permeable membrane, meaning that water can move across, no problem, either way, but what determines that is the concentration of the water and of the solute. Let's watch this video.
Osmosis is the movement of water across a selectively permeable membrane from a compartment where the solute concentration is lower to one where it is higher. The osmotic pressure of a solution is called tonicity. In cell biology, tonicity is determined by the relative concentrations of osmotically active solutes, dissolved molecules, such as sodium chloride, NaCl, inside and outside the cell. Thus, three ranges of tonicity are defined for solutions with reference to cells: isotonic, also known as normal saline, hypertonic, and hypotonic. Since the relative tonicities of intracellular and extracellular fluids affect the osmotic movement of water across the plasma membrane, cells may gain or lose volume depending on the tonicity of the extracellular environment. An isotonic solution, such as normal saline, 0.9% sodium chloride, has the same concentration of osmotically active solutes as the intracellular fluid. For example, erythrocytes placed in an isotonic solution will show neither a net gain nor a net loss of water. Thus, they will maintain their normal biconcave shape. A hypertonic solution, such as 3% saline, has a higher concentration of sodium chloride than the intracellular fluid. This results in the net loss of water from the cell. Therefore, erythrocytes placed in a hypertonic solution lose water and shrivel, a process called crenation. While they lose volume, their plasma membranes have the same surface area, which accounts for their wrinkled appearance. A hypotonic solution, such as water, has a lower concentration of sodium chloride. The higher solute concentration of the intracellular fluid creates an osmotic gradient that draws water into the cell. The osmotic pressure of water entering the cell causes it to swell or even burst, lyse. Erythrocytes placed in a hypotonic solution gain water and expand until they develop leaks or rupture completely in a process called hemolysis.
Okay, we are going to actually spend some time in lab coming up in a few weeks where we're going to be subjecting red blood cells to different salt solutions and looking to see whether the cells lose water and crenate, or whether they gain water and lyse, or whether there's no net influx or efflux of water and they maintain their normal biconcave shape. So we're going to, again, review this more coming up in a couple weeks in lab, but at least you have been introduced to this idea of isotonic, hypertonic, and hypotonic. So good to kind of study this a little bit, and then we will review it again and talk more about it in lab in a couple weeks. This next topic, I am not going to really lecture too much on. I'd like you just to kind of read over this passive mechanism called filtration. So again, I'm not going to talk about it. I would just ask you to please look that over. Now we're going to shift gears and talk about mechanisms that do cost the cell ATP, cost the cell energy to move these substances across the membrane. And the first of them is this term, referred to as active transport. Now, active transport is really the dual opposite of diffusion. Diffusion, remember, was the movement of substances from high to low concentration. Here, as the definition implies, is the movement of substances across the membrane from low to high concentration. So instead of going with the concentration gradient, high to low, we're going against the concentration gradient. I can liken this in some way to a salmon swimming upstream to its natal stretch of water where it was born many years prior. You've all seen salmon coming from the ocean, this big, large salmon, and have to jump waterfalls and get past bears that are trying to eat them, right? They're headed toward that stretch of that river or that stream where they were born maybe 5 or 6 or 7 years ago. It's amazing. They can home in on the olfactory chemistry of the specific stretch of water that they were born in. It's amazing. So as they swim up against the current of water coming down the cascading waterfall, they have to expend energy, don't they, to make those big leaps up to higher pools of water. So in the same way that the salmon is swimming against the concentration gradient, so too would we be moving materials against the concentration gradient in active transport. Now, does the fish have to expend energy to swim against those waterfalls? The movement of the water over the fall. Answer, of course, is yes. So this also costs the cell energy to move the substance across or against the concentration gradient. This mechanism, like facilitated diffusion, is going to employ special transmembrane integral proteins to move the substance from a lower concentration to a higher concentration with the help of energy. You have to have carrier proteins for this to occur. And as we've just said, this costs the cell ATP. When we talk about certain materials moving against the concentration gradient, we're talking about things like charged elements, charged ions. And you see those listed here. Here's calcium, hydrogen ion, this is sodium, this is potassium. These are examples of substances that cells will transport through their cell membrane, again, against the concentration gradient. And here we see the bilayer. Here's one of those transport proteins. And in a very similar sort of way, this particular protein is accepting, because it has a particular binding site, this particular particle or ion. But notice again, it's in higher concentration outside the cell than inside the cell. Yet the cell is pumping it out. It's going against the concentration gradient. That, remember, not only employs the transport protein to help get that particle out of the cell, but the cell has to expend energy to pump it out. So let's watch a short YouTube video that talks a little bit more about this mechanism.
Grammarly does more than catch errors. With Grammarly, you can
Recall that there are two types of cellular transport, passive and active. Passive requires no added energy, but active does require added energy. Active transport also moves from an area of low concentration to an area of high concentration or up the concentration gradient. This is the opposite of passive transport, and because it's more difficult, it needs the energy molecule called ATP, which stands for adenosine triphosphate, because there's an adenosine molecule and 3 phosphate molecules. There are 3 types of active transport: membrane pumps, endocytosis, and exocytosis. Let's look at membrane pumps first. Membrane pumps are just carrier proteins that move substances from areas of low concentration to areas of high concentration. Let's look at the sodium-potassium pump to see how it works. Outside the cell, there's a high concentration of sodium ion, or Na+. The brackets represent the word concentration. Inside the cell, there's a high concentration of potassium ion, or K+. The cell has ion channels that will allow these ions to diffuse and reach equilibrium, but the cell doesn't actually want that. It wants to keep a difference in concentration. So the sodium-potassium pump will force more sodium out and more potassium in all the time. Let's see how this works. First, 3 sodium ions from the cytosol bind to the protein, and a phosphate group from ATP breaks off in the carrier protein. This provides energy for the shape of the protein to change, and the sodium ions can be released into the outside of the cell. Now 2 potassium ions will enter the pump, which causes the phosphate to break off and return the pump to its original shape and release the potassium into the cytosol. Again, sodium will diffuse into the cell on its own, and potassium will diffuse out. The pump forces the sodium to be outside the cell and the potassium to be inside the cell. Another way that cells do active transport is through vesicle movement, either endocytosis or exocytosis. Endocytosis brings items into the cell, and exocytosis pushes them out of the cell. Endocytosis is the process by which cells ingest external fluid, macromolecules, or other large particles, even other cells. We can be more specific about what materials undergo endocytosis. Phagocytosis is where large particles or whole cells are ingested, and penocytosis is when solutes or fluids are ingested. You can think of phagocytosis as cell eating and penocytosis as cell drinking. Here you can see the way it works. First, the cell membrane begins to pinch around the substances, and eventually it forms a vesicle, which can carry the substance to any part of the cell. Exocytosis is basically the opposite of endocytosis. It's a process by which a substance is released from a cell through a vesicle that transports it to the cell surface and fuses with the cell membrane. A vesicle can merge with the cell membrane because they're both made of phospholipids. The layers press into each other, and the phospholipids rearrange a little so that they can open up their contents to the outside of the cell. Again, this requires energy, so it's classified as one of the types of active transport. Thanks for watching this episode of Teacher's Pet. Don't forget to like and subscribe and follow me on Twitter @SciencePet.
We're going to talk about endo and exocytosis in just a second. But that was just a nice review, I think, of the basic mechanism. Again, takes energy to move these materials. Okay, so let's talk about endo and exocytosis, as the video sort of alluded to. Endocytosis and exocytosis both require energy, which in this particular instance is bringing a substance into the cell from the outside. We're not talking about a small little molecule or an ion. Usually this is a bigger substance of some sort. And we're going to describe a couple of different types of endocytosis, two of which the video talked about. Here we have cell drinking or penocytosis. So to orient you, here's our cell, here's the cell membrane, here's the outside of the cell in light blue. When a cell undergoes penocytosis, it's bringing in fluids from the outside. We sometimes call this extracellular fluid as opposed to intracellular fluid. Intra meaning within. This is extracellular. It's outside the cell. And so what occurs is the cell membrane sort of pinches inward, like we see here, and a vesicle is formed. Here's our fluid-filled vesicle, which is then utilized in some manner by the cell. That takes energy to form this little fluid-filled vesicle. The vesicle itself, if you were to examine the chemistry of it, the outer part of it, is the same chemistry as the cell membrane because it was formed from the cell membrane. So you'd see phospholipid bilayer here and embedded proteins as this outer skin, if you will, of the vesicle. But we're, again, focusing on the contents of the vesicle, which is fluid. So we use the term penocytosis. Phagocytosis is literally translated cell eating. So we have, again, a cell. We have some sort of particle that we want to bring into the cell. And we could use the example of, say, this is a white blood cell, and this is a bacterium, let's say, that needs to be removed from the bloodstream, for example, or from the tissues. So what the cell is going to do is it's going to invaginate a little bit, form a vesicle, but instead of having just water or fluid, like we had a minute ago, this particular vesicle, of course, is carrying our phagocytized particle, which will probably then be digested and degraded chemically by the cell. If it was a bacterium, it would be basically neutralized or destroyed by the white blood cell. This, like penocytosis, requires energy. This third mechanism, which the video did not talk about, but this is extremely important as well, is making use of specific receptors on the surface of the cell membrane. And this involves the formation of a vesicle. So it involves the invagination of the cell membrane, just like we saw in the previous two examples. But look at what that vesicle is carrying. It is transporting specific substances that will fit into the receptor sites that were initially found here on the surface of the cell membrane. Okay, So the green guys and the yellow guys here, again, we'll assume there's some kind of molecule. Can they fit into the receptor sites shown here in purple? The answer, of course, is no. The red shaped molecules, however, do have the proper shape so that they can fit into the receptor site, kind of like a lock and key analogy. And that has occurred here. And now we have that invagination, the formation of a vesicle, but that vesicle is carrying a specific special cargo. Again, from the outside of the cell to the inside of the cell, whose specificity was determined by the shape of the receptors, which are proteins that are facing the outside of the cell. Exocytosis, like endocytosis, requires ATP expenditure by the cell, but instead of bringing materials in, that's endocytosis, we are now removing materials from the cell. Exocytosis. Just think of the word exit, maybe. So this is release of substances by the cell. Now, here we have on the left-hand side a recently phagocytized particle, let's say, food, if you will, that the cell is going to break down. And in order to do that, it's going to make use of another organelle, which you will have learned about already in chapter 3, the lysosome. This is an organelle that contains powerful hydrolytic enzymes. And so this lysosome basically merges with the vesicle, as we see here, and these enzymes are going to begin to break down that phagocytized particle. Some of the nutrients that are degraded or produced, I should say, as a result of that chemical degradation might be utilized by the cell in some way, okay? But what about the end products that the cell does not want, that it can't make use of? Maybe they're toxic waste products. We have to get rid of those substances, and that's what's taking place here. That vesicle containing the undesirable byproducts of that digestive process then moves toward the cell membrane, merges with it, and expels the residue. Note what happens to the outer vesicle. It becomes part of the cell membrane. Isn't that interesting? So that's exocytosis. And your book gives an example of this as it relates to the formation of what are called neurotransmitters. If you've ever studied nerve impulse conduction or studied the brain, I'm sure you've heard of things like serotonin, dopamine. These are examples of neurotransmitters that are important in regulating nerve impulses from one neuron, one nerve cell to another nerve cell. And so the formation of those neurotransmitters ultimately begins within the cell, and the cell will then export that neurotransmitter for use in some way by the body. Let's see if we can watch this animation.
The substances used as fuel by single-celled organisms include other smaller cells, particles of organic material, and large molecules that cannot pass through the plasma membrane. Many single-celled eukaryotes use a mechanism called endocytosis to ingest such food particles. In this process, the plasma membrane surrounds and engulfs the food particle. Cells use 3 basic types of endocytosis depending on the size and nature of the material to be ingested: phagocytosis, pinocytosis, and receptor-mediated endocytosis. If the material the cell takes in is particulate, such as a bacterial cell or an organic fragment, the process is called phagocytosis. If the material is a liquid, it is called pinocytosis. Some types of molecules, such as low-density lipoproteins, LDLs, are transported across the plasma membrane by receptor-mediated endocytosis. These molecules first bind to specific receptors embedded in the plasma membrane. The receptor molecules are concentrated in an indented pit coated by the protein clathrin. When sufficient target molecules accumulate in the coated pit, The pit deepens, seals, and is incorporated into the cell as a coated vesicle. Exocytosis is the reverse of endocytosis. This process results in the discharge of materials from membrane-bound packages that migrate to the inner surface of the plasma membrane, fuse with the membrane, and then release their contents to the outside of the cell.
Okay, a nice review of endo and exocytosis. The last thing I'm going to leave you with in this section of chapter 3 is an additional process referred to as transcytosis. And like the filtration section that we mentioned a few moments ago, I would like you to read over this section and just get a sense of what this process is. It's basically talking here about the movement of materials into or across a layer of cells. So I just would like you to please just read over that short little paragraph or two in chapter 3. Okay, that concludes this particular lecture.